Hi there, and welcome to a video outlining one of the major roles of interferon. Now, interferon is produced by the host cell, made by the host cell. And it has a lot of different functions, stimulating the immune system. I'm going to focus here on the antiviral production, protein production. So when the initial host cell is infected by a virus, and I'm going to draw kind of a naked icosahedral virus coming in here for the viral types. So it comes in, virus is making copies of itself, goes through adsorption, penetration and uncoding, then synthesis and replication. Here's the nucleus of our cell. And then ultimately copies are made and released through various mechanisms. Oops. Okay, so we have synthesis, assembly, release. At the same time in the host cell, because this virus is coming in, there's actually a set of signals that are going on inside. So during multiplication, our cell will start making what's a substance called interferon. And I think I mentioned that there are different types, alpha, beta, and gamma. I'm not going to focus on the different types here. We're just going to focus on this kind of central interferon process here. It will release interferon out into the external cell environment. So here's the interferon coming out here. So this is all interferon molecules. And then it doesn't do anything to help this cell, which I think is kind of interesting. But what will happen is a neighboring cell, it has to be a cell that's in the area. There's our nucleus again. We'll pick up this interferon signal, and it's basically an alarm system to let the neighboring cells know, so any cells in the area, that there is a virus that's invading the cells, and to kind of switch on this set of proteins called antiviral proteins, AVPs, and AVP stands for antiviral protein. And these antiviral proteins will then be released within the cell, and as this virus is coming in, it will actually block viral multiplication. But the cell does not know to make these unless it picks up that signal from the neighboring cell. So this cell isn't saved, which I think is kind of interesting, but its neighbors are. So it's kind of not necessarily a self-sacrifice, but a way to, help everyone else out, even though it's not going to do you any good there. So we'll actually block that. And one of the things to always remember here is that interferon itself is strictly, it's just a signal only. It doesn't attack the viruses. It's actually the antiviral proteins made by other cells. So just very quickly to recap, the host cell that's being infected by the virus will have changes in its genetics where it will start synthesizing, transcribing, and translating interferon. It'll be released, picked up by neighboring cells, which if you think about viral multiplication, that would make sense. It's going to be kind of moving around to these neighboring areas. The neighboring cells that pick up the interferon signal will then change their gene expression to start making these antiviral proteins, which will then block viral multiplication. So that's just kind of, it's considered non-specific because it's not acting on a specific virus. It's just a generalized antiviral response, which is really wonderful to think as your B and T cells are getting mobilized for maybe a specific virus, you still have this really important piece here that's already kind of just cutting down the sheer numbers of viral copies that are made. Okay, that's all I have on interferon. Please contact me with any questions you may have. This is Corey Andres at Central New Mexico Community College using